analysis. And I couldn't always have someone as useful as Sean or Al before Sean to do this. So I had to learn a little bit, and that I enjoyed it. And I went on, and now I'm kind of 60-40, doing a bit bench and a little bit of analysis as well. So today I'm going to be talking about the functional analysis of gene sets, because this is something that keeps coming again and again. And there are a number of ways to do this. Of course, what I'm going to show here is what I'm currently finding more useful. It doesn't mean that it's the absolutely best. So I'll be very interested to hear from anybody else who has other ideas of how to do these things better or even differently. Sometimes you get different things from different approaches. So today we're going to be talking about the identification of represented functional categories in a set of genes using software called gProfiler. I'm going to be talking a little bit about ways to visualize the results and summarize them. And very importantly, this is the part I'm more interested in right now, which is reducing the result lists. Because quite often you get hundreds and hundreds of categories, and you need to find a way to summarize this in a matchable form, especially if you want to summarize it in a figure. And I'm going to talk a little bit about an alternative approach using something called gene set enrichment analysis and why is it different from other approaches. So, this is where the problem starts. We do a lot of different kinds of experiments. It could be something simple as just RNA-seq between two conditions, or it could come from something more complicated like a time series when you end up looking at different groups of genes. But at the end, it comes down to, okay, I have a group of interesting genes that were identified somehow. And what do they do? So I'm going to start with gProfiler. So gProfiler is available as a package in R, and it's also available as a web tool. The R package, of course, is very, very useful, and you can incorporate into your workflows. But I find the web tool actually quite useful because you get everything in one go, and you can explore a little bit the data a little bit easier than just using the R package. So I'm going to be mentioning the web tool. The web tool contains 4 modules. The main module is functional profiling, but it does other things that sometimes can become useful. There are other ways to do this, but has the gene ID conversion, which can convert between all kinds of gene IDs. You can do that with Biomart or other packages, but this is right there on the web browser very easily. You can also search for orthologous for genes between different species. And if you're looking into SNPs, you can find SNP IDs and find what genes they correspond to using this other module. It also contains archive versions of both the software and the associated databases. So it allows to reproducibly perform an analysis and always go back to it years later even. What do you need for it? So as input, you need a set of gene IDs. It doesn't matter what kind of IDs. gProfiler works internally with ensemble IDs, but it will internally convert between IDs. And it can be mixed as well. And you need to input to decide what organism you're using. A word about the set of gene IDs. Although it works with ensemble IDs, I actually like to give it here the gene names. Why? Because the output is going to use the IDs that you provide. So it just makes it a bit easier when I look at the gene names. But that's just me. It has a number of advanced options, which I'm going to show you briefly. So the evidence code is actually showing a table of the different categories and the different genes that are in each category. So it allows you to see what genes were in each category, in what categories, and so on. The statistical domain scope, which is the universe applicable to the experiment, meaning if you do an RNA experiment, the universe is going to be all of the genes typically available in the genome based on whatever genome file you use. And that would be the default for gProfiler. But if you have another experiment that the background is not going to be all of the genes, it's just going to be a subset of genes, then you can define it here. And then you can define also the statistical thresholds for the results. And what kind of data you get from here? Well, gProfiler accesses a number of databases. You have the gene ontologies, you have the biological process, molecular function, and cellular compartments. But you also have things like Reactome, weak pathways, and KEGG. You also have some results based on the transcriptional factors. So if you have a bunch of genes that they all seem to be regulated by a particular transcription factor, it's going to show up here. However, this doesn't work so well. It doesn't work so well purely because the association between transcription factor and genes is not perfect. It's not very good at the moment. And you have the same thing with the microRNAs. You also have some other interesting, potentially interesting, they're interesting for me, databases like the Human Protein Atlas, Corum, which deals with the complexes, and in particular the human phenotype ontology. So if you're working with something related to human disease, is a quite useful thing to have. So I'm going to move on to my web browser to show you a little bit how this works.
I'm just downstairs, just finishing up.
Okay. So, can you see my web browser? Someone?
Yep, that's good.
Okay, brilliant. So this is the main page of the gProfiler. So here's where you put your list of genes. And I already input a list of genes here, like I said, just names works. You input what's your organism. The advanced options that you get is, for instance, you can decide to show absolutely all results without any kind of thresholding. Evidence codes or no evidence codes, that's the difference between showing you the genes or not. So I'm going to leave it always unticked. But sometimes when you have long lists, it takes a long time to calculate. So it might be, in that case, useful to leave it out. And you can use here the thresholding for the filtering. Data sources that you want to look at, you have by default all of them ticked. By default, you will only see the ones that have some hits that are enriched statistically. If you're only interested in one or two databases, then it's a good idea to just untick all the others because the process will be faster. So when you're ready, you just run the query. The first thing you're going to find is sometimes there are some ideas, especially if you give names. If you give the gene IDs, you won't have this issue. But if you give the gene names, sometimes you get a gene name associated with 2 or more IDs. So typically I just select for the one that's more informative. And you can rerun it again. And that's it. So this is what I meant by the evidence codes. If you untick the evidence codes and say, don't show them, then you will not have this information, but you'll have everything else. So this is a quick overview of what you get based by divided by database. So you have the molecular function, you get the biological process, and all is ranked by the p-values. Okay. So, all right. So once you have this, you could just export the data by clicking on the CSV, and there you go. And the file will be there. And that's the kind of input that you get. I'm gonna go back to my PowerPoint. So that's the input that I just showed you. You get that what you get here. I personally like to add some additional information because I use that to rank categories later. And the kind of information that I add is, well, first I'll tell you what we get. So first of all, in the first column, we get the source that tells you what database was used, the name for the terms, the ID for the terms. Then you have your p-value, so you get the adjusted p-value. In this, by default, is the Benjamini-Hochberg correction, so it's a false discovery rate. So this is the minus log 10 of the value, which is typically used for graphing, because that would be a bit harder. You have how many genes there are in each term. So there's 12 genes that are involved in this 2 to 5 polyvaginylate synthesis activity. The query size, how many genes of your list did have IDs in this database. And the intersection size, this is how many genes were detected from your set within this category. The effective gene size is, tells you essentially the size of a universe for this particular database. And then you get intersections, which is which genes were detected in your set, in the set that you produce as input, corresponding to each category. So what I like to add to this is the expected number of genes, which is not given, so I can calculate the observers as expected. Although lately I'm finding that it's more interesting to have simply just the percentage of genes identified per term. And I like to add also things like the expression values for all these genes and so on, so I can use them for ranking. So this is the kind of modified version that I have. So we have I calculated how many genes you were expecting, the ratio of observers expected, the percentage of genes in the term, and then here is just, in this particular case, I have the log2 fold change for the conditions that I was measuring, and I also have the indices for because the way I store the data tends to be a collection of tables. They're all in the same order of genes. So what I have is just identify what rows these genes are in. So if I want to retrieve some other data from these tables, I can just come here and easily extract it and do more plots or whatever I want. So how do we visualize this? So a simple way to do that is, okay, maybe rank it by p-values. So the lowest p-values on the top, highest p-values on the bottom. However, I'm not quite sure that this way is very informative because once you've done the initial filtering and you say, okay, anything with FTR lower than 5%, I'm going to take it as an enriched category. So I have a list of categories, but whether one category is 10 to minus 7 and the next one is 10 to minus 6, I'm not sure that's a good way to rank them or meaningful way to rank it. I prefer to rank it by percentage of genes identified in each term, because this seems to be a bit more intuitive what it means. For instance, you have the top class here is the histone H3R26 tetrahydrogenation, which appears very close to the bottom here. The reason it appears very close to the bottom and very close to the top here is that we detected all of the genes, actually 100%. We detected all of the genes in this category as being upregulated in this case. However, there are few genes. So because there are few genes, the statistics are not going to be as strong. So that's the reason it looks like that. And I'm favoring that. Another way that I sometimes use to visualize these things is using box plots. And this particular example doesn't look so impressive because it was a small set that I chose on purpose because it was small and it was easy to show everything on one screen. And a lot of the genes were the same, really. So it doesn't vary all that much. And so this box plot what shows here is the average, well, the distribution of the log2 fold changes in the genes in each category. You can, of course, add things to it. For instance, you can color it by the percentage of genes that were identified in each category. So you can add quite a lot of additional information in this type of plot without taking up much space. And this is one example of a real example where I had identified three different groups in a set of patients of Emery-Dreifuss muscular dystrophy. And we found that our set of patients were they could divide into 3 different groups that had very sharp differences in terms of the genes that were misregulated there compared to the healthy controls. So I used this method using the box plots to visualize in just a single figure to summarize the functional results for the 3 groups I was seeing. So this indicates the genes that are regulated, categories of genes that were regulated, unregulated, so on. So it allows you to condense everything quite neatly in one group. There's another way that I've seen people doing this kind of plots, and I'll illustrate that when I show you the Revigo. This is nothing to do with functional analysis. This is something I put in here because for this plot, sometimes I use transparency rather than gradients. And I was talking to people recently, and they didn't realize that you could use transparencies in using R-based, just basic R functions. Most people tend to associate ggplots with the use of transparencies, but you can use the package scales, which has an alpha function. And all you have to do is define the color and the level of alpha, the level of transparency, and there you go. So with this code here, I generate the different levels of transparency, different colors as an example. It comes quite handy. Anyway, so I've shown you how to get a set of enriched categories in a set of genes, in a group of genes. I'll show you some ways of representing it, but this was a very, very short list of categories. Typically, I get much larger categories. You can have thousands of genes sometimes, and you can get lots of overrepresented categories. And then when you look at them, quite often, many are very redundant. So how do we reduce the redundancy so we can condense the results? So there are several ways to do this. I've come across various packages that claim to do this, but I was never really satisfied with any. So I was doing my own version, which I'm going to show you next to this, because recently I came across this, and I think it does it quite nicely. The only caveat is it only works for gene ontology, so biological process, molecular function, and cellular components. So this is called Revigo, and it's available as a web tool only at the moment. There's an R package in preparation. There should be, I heard about being ready this year, but I don't know. And how does this work? So what it does is it calculates the similarity between Go terms using one of several metrics that are available in the package. Then it uses that similarity to group the most similar categories together, and it chooses a representative category for each group based on a dispensability index that it calculates. And the output is both graphic and tabular. So you get some graphic representation, and you can export the R code that produces those graphs, so you can easily modify them to taste. So that's quite nice. And of course, you have the table, which you can extract and treat as normal. So this is the main page of Revigo, but I'm going to move on to my web browser again to show you live what it does. So that's the page that I showed you. What you need as an input is a list of Go IDs. In this case, I have Go IDs and a column of numbers. So the column of numbers could be whatever metrics that you think could be useful for your analysis. You could use p-values, or in this case, I have here the percentage of genes identified for each category. You have to define, you have only at the moment 4 levels. I think in the R package is supposed to give you much wider scope. But right at the moment, you decide how big you want the list, or how much you want to reduce things. The default is this medium, which seems to work quite well. When you have very, very many categories, sometimes I bring it a bit smaller, but then you do lose sometimes some categories. It's worth, like with everything that is calculated for you, it's always worth checking that it makes sense biologically, and you're not leaving something out of a mistake. If you add some numbers here, you need to indicate what it is. So in my case, it's higher value. The more genes were in the category is better. So I put it there. The other thing to indicate is what kind of metric you want to use for the similarity. The default works really well, but you can play with the others as well. And you can extract the similarity matrices if you wanted to go a little bit deeper into this. So you can use the different methods and compare them. So anyway, all it needs is the co-IDs and optionally this column of numbers. Then you start Revigo, away you go. It's really fast. So this is the other type of representation that some people use. I personally don't like it because it doesn't seem to provide much information compared to the box plots of or the bar plots that I have. But if you like it, it's a way to do it. You can export the R script here, so you can reproduce this graph or change it to whatever you like. And you can export the data as a tab delimited text file. You can view the table here. And here you can see what is done to reduce the list. So you look at the uniqueness and specificity score. And if you remember, I can see it here. No. So I had selected the dispensability score of 0.7, which was the default. And that corresponds to this. So anything that is below 0.7 is going to come up here. Let's look at one group here. See? So for this group, it was estimated that they all clustered together, and it shows this element as representative for a whole group. If you don't agree with this, you can play with it by clicking and unclicking and so on. I don't deal with this here. There's other things that you can play with a little bit. I don't find all this that useful. This is actually quite cool because it gives you a representation of how different Go terms relate to each other. There are things like these maps or tag clouds. I again personally don't like them. There are better ways to do this, but it's there if you want. So what I do is I export the table. And I'm gonna go back to my main one. There. So this is a similar table of what I just showed you. So for one particular group, in this case, he selected this as one group and selected IP10 production as being the representative. So you can just scan through the table, you find the groups, this tells you what it belongs to and whether it was eliminated or not. And these are the principal component coordinates that you can use to reproduce the plot that was shown earlier as well. So all you need is really this table, and you can just generate similar plots and just keep the reduced version of your gene list. Now, Revigo only works with gene ontology databases because it uses a pre-calculated similarity coefficient set. But I was using, before I found Revigo, I was using another approach that is kind of generic, but it's very similar actually to Revigo. It's based on obtaining the list of overrepresented categories and the genes within them, like I just did with for gProfiler. And then I will calculate the similarity matrices pairwise. So all you do is take each category and compare it to every other category and calculate this metric. There are, of course, many metrics available. Revigo, in the documentation, it tells you very nicely about how you calculate these other metrics that they were using. I was using GKR because it's simple and it seems to work. And Jacquard is simply defined as if you're comparing two sets, it's defined as the length of intersection divided by the length of a union. So it's a value between 0 and 1 always. Once I have the similarity matrix calculated, I like to plot it as a heat map, and I use hierarchical clustering to cluster together categories that seems to be similar, and that allows me to identify groups. And then I choose a representative for each group based on, and that's the big question, like based on what? You can use it based on hierarchical levels, so perhaps you want to keep the one that's the broader term, or maybe based on how many genes were included in this category, or sometimes you're not quite sure, but if you know what you're experimenting about, you know the biology, that sometimes allows you to make the decisions based on that. So I like Revigo because it's blind. On the other hand, sometimes I don't agree with their decisions. Anyway, whichever way you do, you end up with a reduced list. This is a similarity matrix calculated using Jacquard and displayed as a heat map and cluster. So here you can see visually that there are, for instance, this category here does have nothing much in common with anything else. But there are blocks here that they have a lot in common. So it's a question of deciding what blocks make sense. So in this case, I select, okay, this will be one category, this will be one, this will be blank. Okay, I see this big block that could be divided perhaps into two blocks. So I divide into this block here, there are all these ones, and this one block here, and so on. So I do that for all of them. The only time when I chose more than one representative was when you have large groups. Sometimes it's for looking at it because sometimes they're all related. But in this particular case, we're looking at cytokines. And we had these two cytokines, one that seems to be upregulated, another one seems to be repressed. So choosing just one, it was going to be leaving this one out. So I chose those three. You know, it's a little bit manual action and your personal interpretation. I think as long as you document well what you've done, I think it's fine. Usually when I do these kind of things, I always save all the original unfiltered data. So you can always go back to it. And if somebody doesn't agree, I can just show it and we can discuss it. So applying this reduction based on Revigo or based on my own manually selected, I went down from 30 categories to, can't remember, it's like about 12 or 13 categories. And usually I would just represent the ones in dark blue. So that's one way to do this. Another reason I like to look at the similarity matrices is that sometimes you discover things that you would miss if you're not paying enough attention. This is a similarity matrix from similar experiment where I'm querying only the human phenotype ontology. So this contains it's a database of human conditions, human diseases, and the genes that are associated with them. So if I use that using every gene that was included in each category, I have this map. Okay, so I can see that generally they're all distinct enough. There's a few of them that have a lot in common. And when you look at this, you can see, okay, this seems to be related to mitochondria. That's related to mitochondria. That could be mitochondria too. So like, okay. This is when you look at all of the genes in the category. But if you do the same, looking at only the genes that were identified in each category, then you sometimes get something like this, which you find it's like, okay, all of these categories essentially were picked up because they all contain various numbers of genes, but they all share a core of genes that are mitochondrial genes. And those were the ones that appear to be misregulated in this particular condition. So when I looked into it, when I saw this, I went like, okay, so what do they have in common? The mitochondrial genes happen to be involved in a complex one, so we're a major source of reactive oxygen species, which was very, very relevant for the biological question I had at the time. And I would have not noticed this if I hadn't done this kind of analysis. So it's worth doing. And this is just a brief example. I just want to leave it as a documentation that using different metrics you can get slightly different groupings, and some work better than others. So it's worth playing a little bit with the different metrics until you're satisfied with one. Personally, I'm quite happy with the Jacquard. You can modify Jacquard a little bit to give you more or less resolution. So that's what it is. And to finish, I want to talk a little bit about this other approach. It's called gene set enrichment analysis to identify, again, categories that are overrepresented between two conditions. The approach that I showed you before with gProfiler is what most other approaches use, which is they start on a group of interesting genes. They know they belong to a universe of genes. Mostly these days tends to be the whole of the genome. But and then you try to find out, okay, for if this category of genes contains however many, you know how many you would expect, and that's the statistics based on that. Gene set enrichment analysis is very different. First of all, you can use, there's a few R packages that are more than this. The ones I've looked at in more detail were FGSEA. F stands for fast, and it's actually very fast. And this is a cluster profiler which contains a lot of very interesting graphing possibilities. There's also a standalone app, which is very easy to use. And what I like about this, it was similar to the gProfiler standalone, is that once you have your input files ready, you can dump them there, press the button, go away, and then you come back and you have a folder full of files. And then you can spend a lot of time going through all these files. This is what I'm going to show you briefly now. I'm leaving this here as a video tutorials because I find this very easy, very useful. There's this, it's only 3-minute one about cluster profiler. It gives you a very quick overview of what GSEA is in general and how cluster profiler works. And then these other are longer videos. I have this 1.5 hour video that is very complete. It gives you all the background about GSEA in general and talks about these two packages here. There's a video on the standalone app that gives you all the background and gives you a nice demo and talks about the different parameters and what you can do in much more detail than I'm going to be doing here. All I'm going to do is just show you briefly how you operate. GSEA is based on a database of molecular signatures that is available in this place. And it's also available as an R package, which is quite useful if you want to find out, okay, I want to find genes that are involved in a particular category, you can just go there. The database is divided in a number of groups. So for instance, all these are based on position. So if you were querying positional gene sets from your data, you might find that it's like, oh, I find that there's a region in chromosome 9 that seems to be overrepresented and so on. You have all the pathways ones, you have regulatory. There's a lot of immunology and oncogenic database gene sets also in there. So you can check that. And also cell types, if you're interested in something like tissue specificity. The molecular signature database can be easily accessed using this package, available in R. And this is just two brief examples of how to use it to retrieve genes belonging to a particular condition. For instance, I knew the ID that I wanted. It was corresponding to cytokine-mediated signaling pathway. So how do I retrieve all the genes that are in there? So first of all, you can see what the different collections are. And then using this type of command here, which indicate your species, the category based on that, and the subcategory will be here. So it will be, in this particular case, gene ontology, biological process. You can just query a particular ID and you get the number of genes. Sometimes you don't know what the actual ID would be. So let's check what's in there. So here I wanted to do something with mitophagy, and there wasn't something directly with mitophagy, but there is autophagy of the mitochondria. So, okay, I'm gonna like anything that has the word autophagy in it. So it gives me all these hits. So like, okay, got autophagy of the mitochondria. So now that I know this, I can just extract the same and extract all the genes. So this is quite useful if you want to look yourself and compare the genes that you have a particular category versus the rest and all kinds of things. So what's the principle for the gene enrichment analysis? So like I said, instead of starting from a group of differentially expressed genes, here we use all of the genes, because what it does is not looking at what is the probability of picking those particular genes from the larger group. It's just looking at the placement of genes. So what it does is it ranks all of the genes in your list, all of the genes in the genome for an RNA-seq experiment, for instance, and they're ranked by expression, comparing the two conditions. So you'll get something like comparing phenotypes. In this case is A and B. So you go from low expression to high expression in one condition and vice versa. So it's the differential of the expression is what counts. So here you start with genes that are at the top of a list are enriched in one category over the other, and at the bottom of the list, vice versa. So the idea is that a gene set that is not particularly enriched, it will be placed anywhere. It will be placed kind of randomly all over the place, uniformly. But if it's a particular category that is overrepresented very clearly in one of the conditions, it will tend to cluster at the top or at the bottom. Depends on what condition it is. So what gene enrichment analysis does is looking at the position of these genes in the ranked list compared to what will happen if this gene set was random, and gives you a statistic and that. So what do you need for this? So you need to start with two files. One is a file that indicates what are the names and what are the samples that we're adding. So here is like, all this is in the documentation. If you go there, so it's 6 samples. I'm going to be 2 conditions. This number 1 is always a 1. I think from historical reasons, don't change it. Here you indicate the labels of the samples, and then according to the columns, what the samples are. So F2, 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 VALC2, VALC2, VALC2. And then you need to provide the file containing the actual data, and has to have this again for historical reasons have to be that, not change it. Then you get how many genes you have in your set, how many samples, and then the first column contains your gene IDs. The description could be anything you like. Apparently, it doesn't matter. And then you have your 6 columns of data have to be in the same order as they were established there. What kind of data? For this kind of thing, we'll be using normally normalized counts. If it's an RNA-seq, if it's a macroarray experiment, you will use expression values, something along these lines. So the major difference with other approaches is that it looks at the placement of genes. So you need to start with a whole list of genes in the genome. And when you use the standalone app, you download it from the page before, and here's where you indicate the actual data you need. So this is the GCT file, the one that contains all the data, the actual data. You will, first of all, you load it and it'll come here. Then which database you want to query. In this particular case, I've gone for C2 Reactome. All this comes in drop-down menu. So it's very easy. How many permutations you want, because it compares your observed position, and then it does a number of permutations. 1,000 is the default. You can change that. I never have to. You indicate the phenotype level. So what conditions you want to use. In this case, is VALC2 versus my F2. And permutation type, that's quite important because that is a major thing on how GSEA works. Ideally, we will use, the options are phenotype or gene set. So ideally, we will use the phenotype. And what it does is then it starts permutating randomly different samples. However, it does require a large number of replicates for this to work well. I think it was 7 or even 10, I don't remember. In the paper, they describe it. So they recommend that if you don't have enough replicates, and typically I have anything between 3 to 5, then it's best to use the gene set permutation, in which case they take the gene set that you've been querying and just permute it on the genome and compares the positions of that against the position that you've set. Then you have to indicate what annotation you have. So if you have ensemble IDs, look for the scroll down for the one that contains mouse ensemble IDs. You have the option of excluding large or small sets. So sometimes when the gene sets are very large, they really are not that informative because, and there are lots of random ones, well, not random ones, but there are sets called biological process. You're like, well, thank you. That's very useful. So by selecting gene sets that are not very, very big, you tend to get rid of a lot of that stuff. You can also exclude smaller sets. These I don't find quite as useful, but it's there. Then you just indicate what folder you want the results to be saved. And there's a number of other optional parameters you can indicate. They're all documented in the documentation. So check that out. And all you have to do is run it. And it can take a while. It depends on how many genes and depends on, how many gene sets and what you're comparing there. And on that folder, you're gonna end up having a file contain called index.html among a myriad of other files. But this is the one you have to go to. So when you go to that one, it opens something that looks a bit like this, and it tells you, okay, enrichment in this phenotype and the other, you get other parameters about the analysis. If you click it into the enrichment results in HTML format, it opens another tab that gives you the different categories that were overrepresented and various metrics. And you have this other thing about details. If you click on here, it takes you to the molecular signatures database website, and it tells you something about that particular category, how it was defined and what it is. And if you click here, you get some more interesting information like this kind of plot. So this is plot here. This is the enrichment score graph, but over here represents where the genes were located along the ranked list of genes. So the more to the left you are, it will be enriched. It will be enriched on my phenotype BAPSI2. The more to the right you are, they are enriched on the F2 or depleted in BAPSI2. So converse. So you can see that this is my top category here, interferon alpha beta signaling. A lot of the genes are completely depleted from my BAPSI2, and they're all enriched on the other category. So this corresponds to a category that is massively downregulated in my F2 condition. I recommend, when looking at this, not just going by the p-values, but looking also at the graphs, because I find that sometimes, although the p-values look not too bad, this graph is a little bit of a mess. So I would use this to select only the ones that are very, very clear. The reason why the p-values and this doesn't match so well is partly because of my use of the gene set permutations, which are not as good as the phenotype permutations. But if you don't have enough samples, that's what you have to do. And after this table, again, you can export it as tab-delimited file, so you can filter it as usual, and then everything applies that I show you to the gProfiler. You could use Revigo for things based on gene ontologies, or you can use another approach like the one I was using, or some other, to reduce your list and then visualize in whatever way. So that's me for now. If anybody has any questions or some things that can be done better than I've done, I would love to hear. Stop share.
Thanks a lot, Jose. Really informative. Definitely a few new tools there that I'll be checking out as well.
Thank you.
So yeah, we'll open the floor to questions, but I have a few myself first of all. So I've used gProfiler quite a bit with RNA-seq data, and you were mentioning you tend to use just the default for the background for the universe set of genes. And one recommendation I've seen is because the seq in particular does filtering of, it actually removes genes where you don't have enough counts or they couldn't be detected in your experiment, and to use the background as actually whatever you get results for in your DEC. So that reduces the background often from 24,000 genes down to more like 16,000.
Yeah.
And I've found the results using different backgrounds can be very different.
Okay.
So I don't know if you have any experience of sort of playing
around.
No,
I never tried that. It's something that I thought about and thought like, it probably doesn't matter, but I never really actually check it. If you see that there's a lot of it, so I imagine that, I don't know.
Yeah, it's not something I've dug into enough to really figure out.
Yeah.
Where...
It does make sense to restrict it to that. Just the genes that were. Yeah, something to look at.
Yeah, I also find that you get, like you said, the transcription factor thing doesn't work that well because you tend to get, hundreds or even thousands of results.
Yeah, because if you look in the actual, if you go to the actual database, you find like for each transcription factor, possible targets are like thousands. Like I'm pretty sure that this gene, and also vice versa, you get for this gene, you get 5, 6, 7 different transcription factors. It's like just because you have that sequence there doesn't mean the transcription factor is actually gonna go there, because it depends on other factors as well. So sometimes it's useful. I do look at it because sometimes you discover things that you go like, if you know about the biology, you go like, oh, this is interesting. Then I look at it separately, but I don't use that as evidence that I have this. I use it more as a nudge to go in a particular direction and see if there's something else by doing other experiments.
Cool. Yeah, my only other thing was more of a request. If you're sort of willing to share your R code for generating the similarity matrices and
yeah, sure. The reason I don't normally do this kind of thing is because I do them in an ad hoc type of way. Okay. So I can produce a reduced one that will work. But maybe for this particular example, I can give you this particular example. So it will work for this particular example.
Yeah, just an example would be good. That'd be cool.
It's very simple. It's very simple. It's really just that function that I showed you there, intersection divided by the union, calculate for all these categories, and just do a heat map. That's all.
Great.
I'll share it.
Yeah, thanks a lot. Does anyone else have any questions for Jose?
Yeah, so I have a question because you're saying that transcription factor doesn't work, but do you know of any other tool that you would rely on better than this one for identification of transcription factor, taking into account that, yes, I know it's tricky.
Yeah,
analysis
so I've used essentially the same database. So this gProfiler is using the transfact database. You can go to the transfact website and you can do your analysis there to find in a set of genes whether you have a particular transcription factor is going to be overrepresented. But it's the same problem. It's the fact that the database is based too much on predicted.
Oh yes, of course.
So you never know. One thing that is provided in transfact is they give you some metric of how, it's not exactly how reliable the association is, but some associations you have data behind it, you have publications, so you can use those as a more confident data set. So you can filter them that way, but it's still, I never found a good way to do this. So what I do is I get what I get, I look through it. If something makes sense, okay, maybe give me some ideas of something to try later. But
no, sure.
So if you know, if anybody, if you knows, or if you found out in the future a better way, please let me know, because it's been a constant for years for me to find a good transcription factor target database that you can trust.
Yes, it will be very useful, because this is basically like if you see something at the level of transcription factors, you can understand all the consequences of this. Yes.
Exactly, and you may have like hundreds of genes, but if you find that, oh, a particular transcription factor is definitely downregulated, well, all the targets are downregulated, they may be related to that, So you can filter your sets a little bit that way, if the data is reliable.
Great, thanks.
I haven't looked at it for a while, but I think the Jasper database is sort of a subset of Transfac or included.
Jasper, yeah,
Rated.
Jasper works, but I have the same problem with Jasper.
Yeah. Okay. Any more questions?
I have one more question.
Yeah.
Sorry. So a question about GSEA. Have you ever used it for looking at the how you call it? 1 second. For the transcriptional profiling of cells. Yes. So basically, to looking for similarity to different cell types.
No, I haven't. But I have the group. I think it's called C8. Is
yes. This is exactly what I mean.
Exactly.
Like the C8 group.
So it works, but it's not so easy. And the reason it's not so easy, and it's the same with the immunology section as well, is that they don't give you nice categories like the gene ontologies, like upregulation of whatever pathway. No, it just gives you, this comes from a particular study where they compare this and that. And then you need to know what this and that is.
Mm-hmm.
You know, so you need to process the data a little bit more and find out what it actually means. So it's not so easy, but the data are there. I used it a little bit for subsets of muscle cells in muscle, different kinds of, because the muscle contains not just your myotubules and myocytes and so on, but contains a lot of, there's a little bit of adipocytes, there's a lot of immunology, immuno types of cells. So when you have a data set that was derived from, say, from muscle, and you compare it to the data set that you have here, for me, it started making sense. I was finding that there was an overrepresentation of categories that they were leading towards adipogenesis, which I know that in my system is something that is screwed up in my myogenesis system. Sorry, so myogenesis and adipogenesis, they're a little bit, they go against each other. When you promote myogenesis, you have to switch off a lot of adipogenic genes and vice versa. So in the system that I had, a lot of adipogenic genes were abnormally expressed. And using this method, GSEA using the cell type, it was quite clear. The adipogenic flavor of a transcriptome was very clear there.
Mm-hmm.
So it does work, but that only worked because the muscle sets, they were very well annotated. So it was very easy for me to distinguish them. For other things I've tried, I have to spend a lot of time, again, clicking on the categories and find out how they were defined originally. So it's not fast.
Mm-hmm.
So I have a love-hate thing with GSEA. I've done comparisons of using gProfiler and using GSEA as well. And things that were not very clear with gProfiler came very clearly with GSEA, but also vice versa. So I tend to do both. But if both work well, I tend to stick to gProfiler.
Okay.
Because GSEA gives you a lot of data, and I just don't have the time to go through it. That's the, in a way, it's the nightmare that we have these days. We can produce data a lot faster than we can analyze it. So you need to leave something behind somehow.
No, yes, of course. It's just the analysis that is like the analysis step is everlasting.
Mm.
Great, thank you.
Okay.
Any other questions? Okay. Jose, one other thing I wanted to say was in R, I think there are sort of numerous packages that allow you to visualize pathways. If you have a KEGG pathway,
Yeah.
I don't know if you've played around with these at all, and they sort of show you a visual representation of each gene and where it is in the pathway, and you can color that by RNA-seq fold changes, p-values, etc.
So I think that the cluster profiler allows you to do that. I'm not entirely sure. It was one of the reasons I was interested in cluster profiler was the different graphing abilities that it gives you, and I think that was one of them, because I'm very interested in that.
Yeah,
It makes it very easy.
Yeah, I'll look that out and I'll add that to some of the notes from this meeting.
That'd be great.
So yeah, so thanks a lot. If we can all thank Jose for his great presentation and for being brave enough to volunteer as our first guest speaker. Thanks a lot, Jose.
Thank you for having me.
Yeah. So the recording of this meeting will go up on our website and on our YouTube page. We'll also get Jose's slides put up there and just a few other resources regarding what we chatted about today. Like I said, the next meeting is going to be September 15th. So I will advertise that, and I'm hoping to book a room so we can meet in person for that. All right. Thanks, everyone. We'll see you next month.
Bye. Thank you.